lectures. That's fine with me. It would take a lot less time on my part. Okay, so today we are going to go over chapter 12, which is about mitosis. And it's actually called the cell cycle. So I'm going to go to a whiteboard and get that to disappear. And I'm going to actually record now. All right, let me get out of this. Okay, so the first thing that I wanted to talk to you guys about is the cell cycle, which is, there's a nice picture in your book, and the whole chapter is about, the cell cycle is about the cell's whole life. Okay, and I think that's important for us to remember. For each cell, this whole life might be a different amount of time, right? So for some cells, it's going to last days. For other cells, it might last most of your lifetime. And so that's why we call it a cell cycle, because it's a circle, like some of the things that we were talking about last week. And you can notice from your textbook, from the picture of the cell cycle, that most of the time that the cell spends, most of the cell's life, if I were to draw it kind of like a pie chart, most of the cell's life is spent here in what we call G1. And then this phase here, which is called S. And then we have G2. And then this portion, this much smaller portion, is for mitosis, which is the focus of this chapter, and cytokinesis, which is the dividing up of the cytoplasm, more or less. Now, cells spend most of their time here in G1. And altogether, these phases from G1, S, and G2, all of those from there to there are known collectively as interphase. And that's because when people first started looking at cells under the microscope, it didn't look, they couldn't see what was happening during these phases, during G1, S, and G2. The interesting bit, when the cell was actually looking different than it looked most of the time, the interesting bit was here, right? So they called this interphase because it's the time in between, the phase in between the exciting stuff that was going on. Now, these letters stand for different activities that are happening, of course, during these different phases. So I'm just going to label them over here pretty small. G1 actually stands for gap 1, which means gap phase 1, and or first gap. And the two things that are happening in gap 1, or the predominant thing that's happening in gap 1, I should say, is growth to the cell's full size and all sort of its normal activities. Okay, so this is when I like to say the cell's just living its life. If it's a skin cell, it's doing skin cell things, it's making keratin, you know, doing whatever skin cells do, staying bound to the other cells nearby it. If it's a stomach cell, maybe it's producing acid, right? Whatever its main job is in the organism, that's what it's doing. I put in your notes, for example, if it's a mesophyll cell in the leaf of a plant, remember the picture from chapter 10, Journey to the Center of the Leaf, right? This cell would be transporting water and carbon dioxide in, oxygen out, making proteins, conducting non-cyclic photophosphorylation, the Calvin cycle, transporting glucose around, talking to its neighbors through the plasmodesmata, right? Just doing whatever it is that the cell does. And cells stay here until they receive a signal that pushes them, that causes them to start to make proteins so that they can enter what we call the S phase. So I'm going to put this right here, a little red block. That's a checkpoint. So when the cell receives signals, that it's time for it to stop just staying dormant and doing its regular things and move on to S. It has to accumulate several proteins here in order to literally push over that checkpoint. So I'll put the checkpoint in red, because we don't want cells to just be dividing randomly. When the cell gets those signals to enter the S phase, S stands for synthesis. Okay, and the synthesis phase is all about making DNA. It's the synthesis of DNA. You'll see it referred to as replication. That's its sort of most scientific name, DNA replication. You can think of it as the chromosomes duplicating, which we're going to talk more about in a minute. Chromosome duplication. That involves not only copying the DNA, but copying the proteins that the DNA is wrapped around. That's what mostly goes on in S. All of the chromosomes are being duplicated. Now, I can't show you this picture at the same time that we're talking, but if I were to draw a chromosome that looked like this, in the S phase, what would happen? Let's say this is my chromosome 1. This chromosome is going to be duplicated, and that's when it looks kind of like an X, right? Or a 2-part chromosome. After it's duplicated, we have names for the different parts of this, but let's just leave that there for now. G2, as you might have figured out already, stands for gap 2, right? Or second gap phase. And this is preparation. All the proteins are made for the real preparation for the actual cell division, okay? Particularly focus on the organelles here. So all the organelles need to be duplicated or replicated so that some of them can go into each of the new daughter cells. Mitosis, I think there's another checkpoint right there. Let me check real fast. Yeah, there's one here at the end of G2 that tells the cell it's ready to go into mitosis. And then there's one sort of in the middle of mitosis, 2 more checkpoints. If all the proteins accumulate here on the G2 checkpoint and say, yes, yes, we're ready for the cell to divide, then the cell is going to go into mitosis, which, like I said, we're going to spend most of the chapter talking about the differences and the different phases of mitosis. There are 5 of them in there. And the point of mitosis is mostly to divide up the chromosomes so that each new cell has an equal number of chromosomes. And then the cytokinesis is, as I mentioned, to divide up the cytoplasm and the organelles so that we make 2 cells, right? We call them each daughter cells. So we started with a single cell, and then we're going to end up with 2 cells, basically one old one and one new one. Okay, add another whiteboard to this. So I would remind you that cell division and cell duplication, this by mitosis this type of cell division only happens in eukaryotes. Not bacteria. Bacteria, prokaryotes divide differently, and we'll talk about that. It's called binary fission. We'll talk about that at the end. I have a picture to show you. But what we're talking about for this type of cell division is only in eukaryotes. But I would remind, right, that means protists, So algae and amoeba, it means fungus, it means plants, and of course animals. Right? So all of these different types of eukaryotes pretty much all do mitosis pretty much the exact same way. A little bit of difference between plants and animals, but we'll talk about that in a second. So what is the function or the purpose? Or why do cells divide? Right? And there's basically three. And if you have the PowerPoint, or when you look at the PowerPoint, you'll see that there's a slide there. It's figure 12.2, and it shows the three different important reasons that cells divide. And I've also written it out on your notes, what those purposes are. The first one is for asexual reproduction. So that means there is no genetic information exchanged or fused. Asexual. It is for one cell to become one set of genetic information, one set of chromosomes to be duplicated so that this type of organism can make a completely new cell. So they show a picture in your book of an amoeba, and that would be a protist. And so this is a form of reproduction for organisms that are unicellular, for some of them. Okay. Another function or purpose is for growth and development. Right? So for an organism to get larger, to have more cells, clearly you are different in size as well as in features from when you were a zygote. When you were just a fetus or even a child, you've grown taller. Structures have developed that weren't there previously. Oh, what did I write? Complex organisms, multicellular, start as a single cell and grow into large organisms. I wrote, think about humans, chickens, and plants. I don't know why chickens, but maybe I was hungry. Development, right? So different cells develop into different tissues, and those different tissues, of course, have different functions in a multicellular organism, right? And they all develop out of a stem cell or type of set of stem cells. And the third function, purpose, or reason for mitosis or cell division is for tissue renewal. So all cells pretty much have a limited lifespan. And so when they die, they need to be replaced. Right? So things like your skin, think about red blood cells. What did I write down? Oh, I wrote down that red blood cells, for example, live on average for only about 120 days. And an adult human has between 20 and 30 trillion of them. So that means you're making 7 billion new ones an hour. Okay, so that's an example of renewal. Bone is another good example of renewal. Some of you have probably broken bones before, so you know that they repair, right? And they repair because that's basically you're renewing that tissue, bone. And even if your bones aren't broken, they're constantly being what people call remodeled. So they are degrading and reforming bone cells, right? Throughout the whole course of your life. Okay, now the next slide that I would show you would be the chromosome slide. And there's a lot of vocabulary that goes with this. So let's start kind of at the beginning here. So if I were to draw a chromosome like this, right? This center constriction, this is a single chromosome. This constricted part has a name. It's called the centromere. It is not always in the center, but in this picture that I just drew, it is, right? So we write down, you can write it's a constriction. Pinch. Chromosomes are made up of two parts. They're made up of DNA, the double helix, plus they're made up of proteins. Okay, so two pieces, DNA plus proteins. And if we were starting with a cell that had a chromosome number, we say, let's a couple more definitions. We say eukaryotes are what we refer to as diploid. And that means it's abbreviated as 2N, where N equals the number of different chromosomes in the organism or the cell. So I'm going to try to go slowly here so you can digest that. Okay, so eukaryotes are diploid, which means 2 times N, where N equals the number of different chromosomes in an organism. So, for example, you are a human, right? And so N is 23. You have 23 different chromosomes in all your, what we call somatic cells. And that literally means all the cells that are not gametes or sex cells. All your somatic cells are 2N. So they would have 2 times 23. They would have 46 chromosomes. So for a human, 2N equals 46. Many times you'll hear people say humans have 23 chromosomes arranged in pairs, in 22 pairs plus a pair of sex chromosomes, and that's fairly common language, especially in animals, to describe sort of the sex chromosomes as different, right? So in human females, for example, we have 22 pairs of chromosomes that are identical, more or less, and then we have 2 sex chromosomes, which are X and X. And if you are a human male, right, you would have 22 chromosome pairs plus a pair of sex chromosomes, X plus Y. So sometimes you'll see it written that way as well. Now, again, I didn't write that out. Gametes are your sex cells. And gametes, as we're going to see in the next chapter, are always N or haploid. Okay, I'm going to erase over here so I can draw a little bit more on my cell. I'm kind of out of space here because it's a miniature board. So because it would be next to impossible for me, and it would be very time consuming for me to try to draw all 23 different chromosomes during our discussions of mitosis, I'm going to draw a cell that has a chromosome number of 3. N equals 3. So it's going to have 3 different chromosomes, and it's a diploid cell. So there's going to be 6 total chromosomes inside my cell. I would remind you, and I'm enlarging the nucleus on purpose so that I have space to draw all of these. 2N, so 2 copies of each chromosome. So in black here, I'm going to draw chromosome 1, and I have 2 copies of chromosome 1. And then in green, I'm going to make chromosome 2, and I'm trying to make chromosome 2 look a little bit different. And in blue, let's make chromosome 3, and make that one kind of short and stubby like this. Okay, so if I count all these chromosomes, I have 1, 2, 3, 4, 5, 6, right? 1, 2, 3, 4, 5, 6. 2N equals 6, arranged in 3 pairs of identical chromosomes. And I don't have any sex chromosomes. This is actually an onion cell, so it looks like this. Okay, this is in G1 or in the resting phase. When the cell goes into S, which remember is for synthesis, and what we're going to synthesize is the DNA, right, for DNA synthesis. So we're going into S. Now all of these chromosomes need to be duplicated. So duplicate the chromosomes. Okay, so here we go. I'm going to make myself bigger so I have more room to draw. Make my nucleus bigger. So now each copy of chromosome 1 gets duplicated. So now instead of just 2 copies of chromosome 1, do you see I actually have 4 copies of chromosome 1? And I made my chromosomes 2 green, so I'll make it green like this. That's 1 copy of chromosome 2, another copy of chromosome 2. And then my chromosome 3. Notice I'm drawing the centromere as thick because it is what's actually, it's the place where the chromosomes and their copies are joined together. Now there's a little bit of vocabulary we have to learn in addition to centromere. When the chromosome is duplicated like this, we refer to each of these copies as a sister chromatid. It's not very well drawn, but I'll put it here again too. Each one of these is a sister chromatid. We used to call them, when I was in your shoes, we called them daughter chromatids, and then for a while we called them twin, and now we call them sisters. I don't know. This isn't my area of expertise, so I don't know why they changed the names. Now here we are at S, and if we go back to my original picture here, okay, here we are at S. Now we need to divide up those chromosomes. We need to duplicate all the organelles, and then we need to divide up those chromosomes and divide into 2 new cells. Right? So now what's going to happen is we're going to go through mitosis, and mitosis is all about dividing up the chromosomes, and cytokinesis, remember, is dividing up the cytoplasm. So mitosis is to divide up the chromosomes, to separate all of these sister chromatids from each other so that we end up with 2 cells, and each of them have the original number of 6 chromosomes. And this is what most of the rest of the chapter is about. It is about the steps of mitosis. So I'm going to pause here, ask if you guys have any immediate questions before I go on, because it won't make much sense if I keep talking and you've got something kind of nagging on you. I think I'm going to get rid of this guy while you're thinking, or I don't know, I'm sort of getting the sense that sometimes you just hit record and walk away, which is fine. But and there's always tomorrow too. You can ask more questions. And at the end of today, we'll also have some time that we can talk about the any questions you have from the pending doom of Wednesday, of course. Okay, so let me go back to this picture here. And again, if you have the notes, they are they spell out all of the phases and what you need to focus on for each of the phases for mitosis, particularly, right? I'm going to start, I think, by telling you the photographs in your book are an animal cell for figure 12.7. So figure 12.7, this is an animal cell. There's a picture later of a plant cell. This is an animal cell. And what I want you to focus on, if you look in your book, you'll see that underneath each of the phases is a long bulleted list of sort of the key features of that phase. And I would say it would be best if you focused on 3 things. Okay. I would say it would be best if you focus on 3 things. And sometimes these things are going to merge together, so it's going to make it a little bit easier. The first thing is the nucleus, like the physical appearance of the nucleus. Sorry. I'm distracted. Is it present? Are all of its parts present? Is it absent? Okay, so that's the first thing to focus on. The second thing to focus on is what is happening to the chromosomes. Okay, and remember, they're already duplicated. They were duplicated in S. Keep saying that, keep reminding you, they were duplicated in S. And, but focus on what's happening to them now. And the third thing to focus on is what's going on with the spindle fibers. So I want to take a few minutes to talk about the spindle fibers, how they're originated, where they come from, what they're made out of, before we begin talking about the individual phases, okay? Spindle fibers. These are going to be the proteins that are going to attach to the chromosomes and separate the sister chromatids. And in your PowerPoint, after the picture of the chromosome, what you're going to see is I put pictures in from chapter 6 to help remind us some of the features of a microtubule, because spindle fibers are microtubules. And you might remember, right, they're hollow, and they're made out of alpha and beta tubulin subunits, right, joined around each other like this. And they're the biggest of the cytoskeleton fibers, in that they're 25 nanometers in diameter there, right? And one of the key features of a microtubule is that the cell can very easily add alpha and beta tubulin units, which will make those microtubules longer. And it's also easy to break those down. Let's say, break down the alpha and beta tubulin units. Right, so what that means is it's really easy for the cell, of all the fibers, it's really easy for the cell to extend them and then shorten them, okay? So that's why we needed to remind ourselves about microtubules. The other thing we need to remind ourselves about cell structure-wise is the centrioles. And we didn't talk about those a lot in chapter 6 for a reason. I wanted to keep the list as short as possible of structures that you had to know. So, centrioles are labeled as microtubule organizing centers. They are, you'll find them in animal cells and some fungus. They are not present in plant cells. Okay. And they're always in a pair. Right? And your book is going to call that pair, or that combination of a duplicated pair, a centrosome. So they usually sit like this in the cell at 90-degree angle to each other. Like this. Yeah, I guess I should have made them blue, right? Because they're made of microtubules. And what we're going to see is they are a microtubule organizing center. So what that's going to mean is the spindle fibers, which are microtubules, are going to radiate. They're going to grow out of the centrioles at different lengths. That's not a great drawing. It's a better picture in your book. Okay, so again, we're going to keep track of what's happening to, what's happening to the nucleus, what's happening to the chromosomes, and what's happening to the spindle fibers. Okay, so that's what we want to keep an eye on in all of the drawings of mitosis. Now, there are 1, 2, 3, 4, 5 stages plus cytokinesis. So we're going to go through them one by one. Let's see. All right, I'll leave that up there and I'll just write here mitosis. And I would like to draw underneath, or draw and then right underneath. I'm going to draw to the and then right on the right hand side. I think that'll be easier. I can make a bigger picture that way. So the first phase is called prophase. So it's kind of the get ready phase, right? It's the first phase of mitosis. And we're going to stick with our cell that has 3 chromosomes. And I'm going to write out for you in abbreviated form the nucleus, the chromosome, the spindle fibers. Again, it's written out for you in immense detail in the note outline. And I think I put it in as a PDF so you could just open it up and see it at the same time. Okay, so first thing, prophase. So I'll draw my nucleus, so it's still there. But what is different is the nucleolus is gone. So remember the dark spot where the ribosomes organize? That isn't there anymore. Okay, so that dark spot, the nucleolus, I'm going to erase it there. It's gone. Remember, the chromosomes are already duplicated and they are condensed. So if we were keeping track of what we needed to, we might say the nucleolus disappears. The chromosomes, we say condensed, are condensed. Chromosomes, sorry. And by condensed, we mean that we can see them. So we would be able to see chromosome 1 and its partner, and chromosome 1 and its partner, the duplicated chromosomes. I think chromosome 2 was green, I think. All right, so this is chromosome 2, and then the blue one is chromosome 3. Right? So just kind of drawing them rough. Right? So chromosomes 1, 2, and 3. Also, because we need to keep track of what's going on with the spindle fibers, we would say the spindle fibers are starting to lengthen and radiate out of the centrioles, and the centrioles, which are duplicated, are starting to move toward the opposite poles of the cell. So remember, the centrioles are set like this, but they're starting to split apart from each other and head in opposite directions. And I don't have purple, unfortunately, right? So I guess I use red. So if I had purple, right, I would make the spindle fibers purple because they're protein. And you're going to see some of the spindle fibers are going to be still kind of short, and some of them are going to be longer. But the centrioles are starting, the spindle fibers are starting to grow, and the centrioles are starting to migrate apart. Okay, that's pretty much prophase. The second phase we call, pro-metaphase. So this phase wasn't a phase when I was in college, but it is now. And pro-metaphase has some important things in it, which is why it became a phase, some key events that we need to focus on. So I'm going to draw my picture again. Try to make it a little larger this time. Sorry. The first thing that we would notice is that the nuclear membrane, the nucleus, is broken into pieces. So the nuclear membrane, we say, is fragmented. Which, if you think about it for a minute, that makes sense, because if we're going to move the chromosomes out of the nucleus and we're going to separate the newly copied ones from each other, we have to be able to get them out of the nucleus. So the nuclear envelope fragments or breaks into pieces. Okay. The chromosomes, which is also what we're focusing on, right? The chromosomes become even more condensed and shorter, and a new group of proteins assemble on each side of the centromere of each chromosome. And I'm going to draw that large over here, and then just for chromosome 1, and then so I can show you and label all the pieces. So if this is our duplicated chromosome 1, for example, a new set of proteins assemble right here. I shouldn't make them red because I'm making spindle fibers red. And then yellow. Sorry, I'm indecisive. Looking blue because that's the closest thing I have to purple. A new set of proteins, and I'm exaggerating what they look like, assemble on either side of the centromere, and they look like that, like a C or a cup or a connector. And those proteins have a special name, of course. They're called the kinetochore. So they're called, and they assemble at the centromere. Kinetochore assembles at the centromere. And what they are is they are, right? They are a connection place for the spindle fibers. So I'm going to draw an exaggerated spindle fiber here, like this. And I think you can see where I'm going with this, right? Each of those is going to be a connecting place for a spindle fiber. And then when the spindle fibers get shorter, they are going to stay attached at the kinetochore, but they're going to literally pull those sister chromatids apart, which I'll relabel those as sister chromatids to remind you. So that brings us to the key feature of pro-metaphase. The key feature is that the spindle fibers begin to attach to the sister chromatids at the kinetochore. Okay, so I'm going to draw some of my chromosomes here. So in order for that to have happened, the centrioles have to be completely migrated to the opposite poles. So I'm going to draw them here and here. Like that. And the spindle fibers, some of them need to be radiating outward here. And I think I'm just going to draw like chromosome 1 on its side like this. Like that, right? And I'll put the blue kinetochore here. It's not perfect, but I'll put one there. And then maybe I'll give a spindle fiber coming up like this, right? So one copy of chromosome 1, for example, in pro-metaphase has a spindle fiber attached to each of its kinetochores, but maybe the other copy of chromosome 1 doesn't yet. Okay, so the other copy of chromosome 1's over here with its duplicated part and its kinetochore like that. And let's say this guy only has an attached here, but he doesn't have one attached from the north yet, if you want to think of it like that. right? We think of this as the metaphase plate here along the middle, and we call this the poles of the cell for orientation. And I'll put the other chromosomes in here in their duplicated form as well. Oh, there's the green ones, and then we had baby blue ones, right, for chromosome 3. So some of them are going to have spindle fibers attached to them, but not all of them yet. Okay, that's prometaphase. Okay, metaphase. That's black. Metaphase is marked by all nucleus is completely disintegrated, completely gone. I don't know. And the key feature here is going to be all chromatids, all sister chromatids have a spindle fiber attached to their kinetochore. That is the big thing. Right? Red, yellow, whatever. That's the thing that you have to remember about metaphase. So if I were to draw it, and we say all the chromosomes are lined up on the metaphase plate. Remember when I said there was a checkpoint inside mitosis? This is the checkpoint, that all the chromosomes are lined up on the metaphase plate. So I'm going to put chromosome 1 here and the other chromosome 1 over here. Right? And chromosome 2's there. The other one may be right next to it. And then our chromosome 3's, one there, maybe one here in the end. So, okay. And every single one now has spindle fibers attached at their kinetochore. Like that. And they're all lined up, we would say, on the metaphase plate. So I'm going to draw the centrioles up here, which are the, like I said, the spindle fiber organizing centers. But remember, plant cells can do this just fine without centrioles. So I don't know any more information about them than that to tell you. Okay. Key thing to note, see how I didn't draw, I'll put the kinetochores here on each of these while I'm talking. See how I didn't draw all the chromosome pairs together, right? So this is one chromosome 1 set of sister chromatids, and here's the other pair of sister chromatids for chromosome 1. In mitosis, it is important to notice that all of the pairs, all of the duplicated chromosomes are not together. This is going to be different in the next chapter. At metaphase, they're all going to be lined up together. Okay, so that's another important feature. Anaphase sister chromatids shorten, pulling, or sorry, spindle fibers shorten, pulling sister chromatids apart. So here's my chromosome 1, and always in books now they change the way the chromosomes look, the way the sister chromatids look. They kind of look like this now, which I'm not good at drawing. So I'm only going to draw one of them. Okay, maybe 2. So they kind of look like V's or birds, the long ones. Because remember, this was our chromosome 1 pair that's being split up. I'll draw the other chromosome 1 pair. I don't remember where I had them in relation. Over just a little bit. So there would be blue pair, chromosome 3, being pulled apart here. If I could see all my pictures at the same time, which I can't. And then I think I had the 2 green ones kind of together. And some of them are going to be at different places as far as how pulled apart they are. This doesn't happen like a perfect orchestration. So like these green ones might still be close together, but let's say the other pair has already moved. The sister chromatids have already been separated much more like that, right? Then I think I had our other chromosome 1. So I'm going to draw those still kind of almost not, nothing's happened to them yet. Right. And then the other blue pair, I'm going to make them somewhere in the middle. Make those. I usually tell people, try to imagine, you know, the chromosomes are fluid. They're not stiff like a straw or a coffee stir. They are much more like rope, right? So when you pull them apart, I'm going to stop the share for a second and just do this, right? So for example, if these are my chromosomes, this pieces of pink rope that I have, right? So when they're laying on that metaphase plate, they're like this, but when I start to pull them apart, right, these are actually kind of stiff. I should get the other ones, right? They're drawing them like this because only where my fingers are, that's the only place. That's the only place where my fingers are the spindle fibers. So it's the only place they're attached. So when you pull them apart, they're gonna do this. They're gonna form a V, right? So they're gonna look like this. When they're laying on that metaphase plate, they always draw them kind of flat like this. But you can see even, you know, they're rope, but they're not. They could be twisted like that. They're not just perfectly like rigid pens just laying there, right? Okay, let me go back to my whiteboard, son, and they're all gone. Oh, I didn't know that was going to happen. Okay, well, then I think we were on anaphase, right? And the last phase is going to be telophase. And I tell people you can think of that as like reverse prophase. So if you memorize what happens in prophase, telophase is the opposite. So we usually kind of draw it like this because telophase and cytokinesis are kind of going on at the same time. So the nuclear membrane reforms, the nucleolus reappears, the chromosomes relax, and they become long and skinny and looking like, just like, I don't know what, like rope in there. You can't tell one chromosome from the other, right? Looks like that. Spindle fibers are almost completely disintegrated. So we would still have our centrioles on the poles, but there's like barely any spindle fiber sticking out of them on each cell. Let's draw one up here too. Right, so there's barely any spindle fiber sticking out. And then cytokinesis, which there are pictures in your book. It's different for plant cells versus animal cells. In plant cells, what we call a cell plate forms that's made of vesicles that fuse together. So we usually draw plant cells as almost like squares. They tend to be more rectangular in shape. And then literally what happens between the two cells is these little tiny vesicles form like this, and then they start fusing together and get bigger and bigger until they're two separate cells. In animal cells, we call it a cleavage furrow. And it looks like this, more like a pinch. And it's a ring of actin. And I wish I could show you the picture at the same time as I tell you this. It's a ring of actin. You can think of it like a drawstring purse. It pulls tighter and tighter until the two cells are split apart. So it would actually, I should have said microfilaments, right? Okay, that's the end. Technically, that's the end of mitosis. There is a picture, the next picture that I wanted to show you is how bacteria do cell division. And so prokaryotes, they engage in what we call binary fission. Bacteria only have a single chromosome to copy. So usually when I draw it in microbiology, I draw it like this. It's a single chromosome that's a double helix and double-stranded, right? But it's a loop, not individual chromosomes. So the first thing that happens is, just like in plant and animal cells, that's going to get duplicated. I'm going to have two of those rings, and they actually get attached to the inside of the cell membrane. So the phospholipid bilayer should be in here. And then very simply, this cell is usually, we always draw it as a rectangle, but if it's a round cell, it would be round, right? Then we're going to get pinching off of the cytoplasm. And we're going to end up with two bacterial cells. Because there is so much less work to do here, okay? We only have to copy one chromosome, and we don't have any organelles. We just have a few ribosomes in there. And maybe a couple other things, right? This only takes about 20 minutes in most bacterial cells, so the binary fission happens much more quickly, but it is asexual reproduction, right? We're not having two different cells combine here, we're having a single cell form into two. Our cells and cells that undergo mitosis, I think in the picture of the cell cycle, your book says how long approximately. It's different for different cells. So if this is G1, this is S, and G2 again. And then, well, that wasn't big enough, right? Yeah. And this is mitosis and mitosis and cytokinesis. Hours in S and G2. And mitosis itself, I think it varies how long it takes in different cells, but I think at least some of these, like S, might be 6 to 8 hours, I forget. But mitosis itself, maybe just a couple hours. Now, why do we care? Why do we have to learn about this? Right, so you remember from the beginning when I put up these checkpoints, one there, one here at the end of G2, and one at metaphase. The reason that we still are interested, one of the reasons we're still interested in the cell cycle, is because, and like I said, I'll just keep this very short, cells that divide, we would say inappropriately, or divide too frequently, right? Cells that don't follow the cell cycle, that these would be cancer cells, right? These would be tumor cells, because they have lost the ability to stop at the checkpoints, and they just continually go around the cell cycle. We call it uncontrolled growth, right? So that's why people are still interested in the cell cycle. One of the reasons is because if we can understand the checkpoints and we can understand what's wrong with cells that can break down those checkpoints, then in theory, we could prevent them from forming a tumor or causing a growth and causing cancer. So that's kind of the point of that. Okay, I'm going to stop recording now.